and Facts and Snacks, Lunchtime Journal Club discussion. My name is Electra Barrientos, your host and a community engagement specialist at the Research Centers in Minority Institutions, or RCMI, Community Engagement Core, led by Dr. Carla Williams. Our group's primary goal is to ensure that RCMI supported research addresses the needs and interests of the local community. To achieve this, we partner with local organizations to address health-related concerns, promote community participation in research and partnership with researchers in study design and execution, and ensure research findings are shared with relevant communities. The CEC offers consultations to Howard research teams to incorporate community engagement best practices into their projects, as well as facilitate community partnership introductions and more. I am very pleased to welcome Dr. Shaolei Teng as our speaker this afternoon and hear more about his COVID-19 research. Dr. Teng is an associate professor of the Department of Biology and director of the Biology Graduate Program at Howard University. He received his PhD in biochemistry and molecular biology from Clemson University and completed his postdoctoral training in bioinformatics and genomics at Cold Spring Harbor Laboratory in New York. Dr. Teng's research interests are to develop and apply bioinformatics approaches for analyzing the genetic variations associated with human diseases and discovering biological knowledge hidden in the massive data sets. Dr. Teng has published more than 20 papers in peer-reviewed journals, such as Molecular Psychiatry, BMC Genomics, Amino Acids, and Biophysical Journal. Thank you so much for joining us, Dr. Teng.
Okay, Lisa, thank you for the nice introduction. Okay, let me share my slide. Okay, could you see my slide at the mouse?
Yes.
Yes, very good. I can see it.
Okay. Thank you for the nice introduction. So today I want to talk about actually several projects regarding the SARS-CoV-2 and also the mental disorder. Okay, before I talk about the story, let's see some two group numbers. The left picture is the I just get a screen from the Johns Hopkins coronavirus this morning. So the total infection case is already over 570 million. We all know this number is underestimate. So I guess there may be already about like 1 billion cases in the worldwide. And the total death is about the 6 million in the worldwide. We know the coronavirus is like a new disease in these 2 years. Other than the right picture, we have this kind of older disease, the mental disorders. And in the 2017, the total estimate is about for the mental health disorder patient is about 792 million. So these are very two common disease. Of course, there are many factors that could contribute to this disease, like society or economic factors. So in today's talk, I want to focus on the genetic factors for these two diseases. So my talk will include 3 parts. First is psychogenetics. We talk about how is next-generation sequencing to research the different pathway genes. Then the second talk is about the coronavirus itself, how is structure modeling to research the coronavirus spike mutations. it's a human reset proteins. Then the third one will focus on this paper. We talk about the relationship between these two diseases. When we talk about the genetics, we first must know is our human genome. So our genome actually is a very big genome. It includes 3 billion base pairs. I think many students like read the Hilbert books. The total word is about 1 million words. If we say the one word is average 7 characters, then our human genome, if we think one base pair is 1 character, then it could include the information as many as 428 sets of Hilbert books. If we put this book together, then the height is about like 40 to 50 story buildings. So this is the highest, the tallest building in our DC Washington monument. So in our human genome, it includes a lot of information. To get this information, we need to first sequence our human genomes. In the 1990 to 2003, scientists launched Human Genome Project. It took 13 years and spent total $3 billion to sequence the first human genome we call the reference genomes. After that, then we think about sequence the personal genome. So in this picture, the white line is the milestone that represents the normal speed of technology development. Then the green line is the cost per genomes. In 2007, when we sequenced the personal genome of Dr. Venter, he's the pioneer of the human sequencing project. So it still took like 4 years, $100 million. Then the next year, when we had the revolution technology called next generation sequencing in the field, when we sequenced the Dr. Watson personal genome, it just took 4.5 months and get like $1 million to sequence his genomes. And right now, use very powerful sequence machines. We can get our genome in a couple of hours. At the price in theoretical, we already down to like 1,000 per genomes. So this gave our not helpful research the genetic questions. For example, we can sequence thousands or millions the different patient or genomes, then try to identify some gene or mutation associated with this disease. And not just humans. For example, for the virus, I think in the 2003 for the SARS-CoV-1, it took like several months to get the genomes. But for the SARS-CoV-2, when they identify the original virus strain in Wuhan area, and then they just take a couple of days to get the entire genome. Then Pfizer and Moderna can base on the genome and design the vaccines. But this also brings some questions. So in the left picture shows the growth of dbSNP. dbSNP is the mutation database in NCBI. We can see blue line is the submission of this SNP. Once the next generation sequencing in the market, it has we can see submission has a huge increase. Right now, there are like hundreds of millions mutations in this dbSNP, but we know the most of the mutations they are function neutral. Only a few they has a response for some phenotype or maybe some kind of disease. How we identify this kind of important mutations is like find a needle in a haystack. This we call the data-rich information pool. This is why there are many kind of the bioinformatics or biostudy approach to apply to this field. I think the many students from the medical school, they are very hot area we call the precision medicine. Precision medicine based on basically we try to develop some drugs based on the human genetic variations or like a personal FD design some drugs specific for certain peoples. So basically, we can get the sequence from the populations of family members, and then we use next-generation sequence to sequence that. And when we identify the variant, then my job is to apply different computational approach to identify this variant and genes, and then based on this target, we can design some special drugs. Okay, let's go to our first story for the psychiatry genetics. So we know the mental disorder, psychiatric disorders, they are very common disease. It's reported nearly 1 in 5 Americans may have these mental health issues. So here shows the 12 months prevalence in all disorder is about 18.6%. Then for some serious mental disorders such as depression is about 7%, bipolar 2%, and schizophrenia is about 1%. Another reason why this research is so important because the psychiatric disorder actually is ranking #1 for the burden of disease, in almost twice than the second heart disease or third cancers. So each year we spend more than $6 billion to research to fighting this disease. So understand the mechanism of this disease is very important for our society and the economy. Mental disorders that actually they are highly heritable. So the left picture shows the risk for developing schizophrenia could increase by the renaissance. For example, in general population, we just show is 1%. But if a parent have a but if a parent have the schizophrenia, then risk for child could increase to 60%. For adult teens, about 70%, and adult teens is 48%. So when we calculate the heritability, schizophrenia could reach to 70 to 85%. Bipolar is almost in the same level. They are much higher than other complex diseases such as breast cancer or Parkinson's. So this implies that the genetic factors could play very important roles for developing these mental disorders. One of the very important genetic factors is the sequence variation, the mutations. In the earlier of this century, the scientists researched this big Scottish family. Many family members they have the mental disorders. And then they found many of these family members they carry a sequence variation, translocation that is from the chromosome 1 to 11. This translocation could disrupt brain expressed genes. They name this gene as disrupt in schizophrenia 1, this one genes. As later, they found this actually a target gene for many like schizophrenia, bipolar, or depressions. But we know the psychiatric disorders, they are complex disorder. Multiple genes contribute to that. This protein actually is scaffold protein. It could interact with many kind of these other proteins. And this protein play very important roles in the neurosignaling. And also this one could regulate the expression of any other proteins. So these genes are also very important target for the mental disorder. So we for this one pathway we name as a 2 pathway. One is this one interactome, and the other is this one regulon. So we think maybe some genes in this one pathways and some mutation may be responsible for the mental disorders. So then what we did is we use next generation sequencing. We sequenced more than 1,500 samples that include 6,000 psychiatric patients, like each 200 for the schizophrenia, depression, and bipolars. And also we had 889 health controls. Then we use we call the target sequencing. We try to sequence the coding region exon as promoter, as that's very important for the regulate gene expression and also conserve the regions. And then we sequence that and we identify many kinds of sequence variant. So when we identify this sequence variant, we try to want to know how it could associate disease. So in here we apply 2 different biostudy approach to do that. The first is for the common variant, we use like the GWAS study. We want to find some variant that enriched in the psychiatric patient. But the next generation sequencing, one advantage is we can also detect many kinds of real variant that like very specific to the certain peoples. Because this real variant only appear in the few samples, to increase its power, then we try to do association based on the gene levels or gene set level. Gene set level is pathway levels. And we try to look at this real variant in the different function class, and we try to look at its case control and also the quantitative trait analysis. So here is our result. For the common variant, when we apply like GWAS study, we found a variant associated with the major psychiatric disorder in this gene, the neuron axon 1. So this we call the Manhattan plot. Here is each dot is mutations, and then we get their minus log p values. If it is more significant, then they should be more high. When we zoom out these regions, we can see many signals appear to this variant and also its neighboring variant. So this is some target gene we identified. Then for the real variant, we also found some interesting signals. For interaction for this pathway, we didn't find some very significant signals. But for the other pathway, regulon, that is this one could regulate its gene expressions. We found some signal between the disrupted real variant with the schizophrenia, and also it could contribute to the combined psychiatric disorders. Okay, this is we talk about the from next generation sequencing. We talk about the psychiatric genetics. Then the next part, I will specifically talk about the coronavirus, how we use like computational biophysics method to research their proteins. Then I'll give a little background about the coronavirus. Coronavirus, this is a very big virus families. We found 7 coronavirus that could infect the humans, include 4 like endemic coronavirus. They just have very minor symptoms. We also have 3 pathogenic coronavirus that include MERS. The death rate is about 35%, then SARS-CoV-1 about 10%, SARS-CoV-2. The COVID, at the beginning, it's also very high, about 5%, but right now, because of the vaccines, because we have these different drugs, then the mortality rate already reduced below the 0.5%. The different coronavirus, actually, they share very similar genome structures. About like 1/3, they encode some non-structural protein like OIF proteins. Then the remaining 1/3, they instruct some structural protein like amino protein or membrane protein. I think among all these proteins, the most famous one is the spike protein. So we know the coronavirus, they use spike protein. They could interact with human receptors. So spike proteins, they have 2 units, S1, S2. S1, they have the receptor binding domains, RB domains. They use this domain, they could interact with the human receptor. Their binding interface we call receptor binding motif. In the SARS-CoV-1, SARS-CoV-2, they use the human ACE2 as a receptor. MERS, they use different DPP4 as a human receptor. So if we compare these three spike proteins, when we do the sequence alignment, SARS-CoV-1 and 2, they're quite similar, like around 60%, but MERS, their sequence are very different with SARS-CoV-1 and 2. But it's interesting when we do the structural alignment, of course, the SARS-1 and 2 are their IMSD is very low, 0.5, okay? But when we compare the MERS with SARS-1 and SARS-2 is 1.8, it's still below 2. So that indicates actually their structure are they are most similar. Because we know this coronavirus, the RNA virus, they mutate very quickly. At most of this mutation, actually they're no effect, okay, found a neutral effect. But some mutations, they may have very important effect. They may enhance their binding affinity, enhance these transmissions. That because they mutate so quickly, so then bring some challenge for our experiment design. So here we try to use the computational saturation genesis, try to look at the effect of all possible mutations. So basically, for the every amino acid in the proteins, we mutate to other 19 residues. Then we use some sequence-based machine learning to look at their mutation pathogenicity, protein stability, to see if this mutation involved to use the post-translation modifications. And also, I think today I want to focus how we use the structure-based computational biophysics method to look at how this mutation could affect protein stability or protein-protein interactions. This is how we did. So basically, we calculate the difference between the folding energy of the mutant and the wild type. If this mutation is greater than 0, then that means this mutation could destabilize the spike protein or ACT protein stability. Destabilize, then if we get the negative values, then this mutation could have a stabilizing effect. So basically, we calculate all possible like 18,000 mutations in the spike full-length. And for that mutation located in the interface, we also, for this mutation located in interface, we also, for the mutation located interface, we also look at the calculate their binding ion change between the mutant and wild type. If some mutation gets the triple delta G greater than 0, then this mutation could weaken binding affinity. But if some mutation are negative values, then this mutation could potentially increase this binding affinity. We announced about 3,700 mutations in the spike RBD regions, and also another 11,000 mutation in the human ACE2. So here, first we look at how this mutation affects the protein stability. We found the majority of the mutation, about like 30%, they could strongly destabilize the entire spike protein. And about another 30%, they could destabilize the spike protein. We also see the very similar patterns in the RBD regions. Then we calculate their mean values in the of all these mutations for each residue, try to identify some key residues. The line chart shows this residue from the top first, like G431, to range to the top negative as 514. Okay, let's first look at these top two residues. We can see from this heat map, all these mutations in G431, they could decrease the spike protein stability. This we have very similar patterns for the SARS-CoV-1. And also S514 from the sequence, actually, these two mutations are very far away. But from structure, this very close to the G431. So a lot of the mutations in the S514, actually they can make the spike at the RD region more stable. And we can also see very similar patterns for the SARS-CoV-1. We just look at the computation predict ones, and we look at the real mutations, the real viral mutation D164G. So this mutation actually probably is the first dominant mutation in the earlier of the pandemic. When we calculate the delta G stability, we found the G164G is minus 0.784. So this is the most stable form compared to all other possible mutations. And also from the location, we can see this mutation actually very close to the glycylation site N1616. So because we know glycylation is very important for protein stability. So we think maybe this mutation could enhance the fitness of the virus by increasing its stability and also participate with these post-translation modifications. Then we look at how this mutation could affect the virus at the receptor binding affinity. We can see most of this mutation, because it's far away from interface, they had no effect. But some mutations in this receptor binding motif, they have very strong effect. It's also very interesting that when we look at the viral variation, it's about like 9.3 could enhance this binding affinity, but in the computational theoretical predict one only 3.4. So that means the viral mutation during the elution, they could kind of enhance this binding affinity at the survive. So if we look at the top two cases, the G496, so we can see in the SARS-CoV-2, most of the mutations, they could weaken this binding affinity because the glycine is the smallest residue. When we change the large residues, it may make this binding interface unfavorable. Very similar pattern in the SARS-CoV-1. Then it's very interesting is the label residue F497. So we can see many mutations happened in this residues. They could enhance these binding affinities. However, when we look at the SARS-CoV-1, we can see many mutations. They have very neutral effect. This is different between these two viruses. Okay, then let's look at another real mutation, the viral mutation, L452R. L452R, so when we calculate travel delta G bindings, so it's minus 3.95, so it could enhance the virus spike interaction to the human ACE2. So when we do this research, actually this in the earlier pandemic, when we submit paper in the May 2020, at that time, only a few patient variant strand carry this mutation. But then in the early of the last year, so in the California strands, they found this mutation become kind of the dominant mutations. The LA Times, they also report our work. Then until recently, we know the Omicron, they have BA.4 and 5. This sub-variant, they increase the transmission again. And then they found actually L452R, this is quite the marked mutation for this virus strand. So this indicates the travel delta G, this binding unchanged could be very useful, provide very useful information for all these functional mutations in the dominant variant strands. Actually, we apply this kind of approach to the different disease tag mutation. We do not have time, so we also for other coronavirus protein like OS3A or some non-structured proteins, we not just focus on SARS-CoV-2, we also look at the SARS-CoV-1 and the MERS proteins. And for the human receptor, we try to do some experiment, try to design some therapeutic peptide. And we also apply this to other target protein. We even use some do some animal study, like we do the Drosophila to look at their mutant. Actually, the primary data shows some very interesting phenotype, and we apply some kinase for associated with the intellectual disability. And especially, we also use this method to look at our DIS1 pathway genes. So later, I talk about the NRX1. Okay, so I will go to the last part. So what's the relationship between these two diseases? That is, of course, we talk about the genetic factors. So I think in the post-pandemic, we talk about this non-COVID, right? So this non-COVID patient, they have many kind of symptoms, such as they may have some respiratory or heart problems. They may not so smell or taste for a long time. Then they have identified about 1/3 of the COVID patients, they may experience some neuropsychiatric problems, like some depression, anxiety, cognitive deficit, okay, and also other kinds of cognitive issues. So how these two diseases may be associated together, okay? So in this review paper, we systematically reviewed all these many kind of these host factors. So we know, I just showed the coronavirus actually it used on our human receptor. We just show the SARS-CoV-1, 2, they use ACE2, okay, and also the MERS, the DPP4. Actually, there are many other proteins also help to enter the modulate the virus enter to the cell, such as TMPP, PRSS2, furin, okay, ADAM10. Okay, then after that, there are kind of many kind of mechanisms. The most common is inflammations. There are also other kind of cell survival, the endorphin signaling. Then this mechanism, they may implicate some mental disorder like the depression or schizophrenia. So we know the virus enter to cell, they must use some entry proteins. We just show the ACE2. ACE2 actually, some research found that this ACE2 may be involved to some depression or cognitive impairment. And also DPP4, it may relate to some PTSD, other depressions. So and also the furin may they identify some mutation in the associated with furin or BDNF expressions, and it may relate to the Alzheimer or schizophrenia disease. Then after the virus into the cell use this receptor proteins, and some mechanism could activate by these infections. For example, we know the most very known is the cytokine storms, right? Cytokine storms that could lead to some suppression of the B cells and also antibody productions. Actually, this GSK3 beta. So GSK3 beta, they found in the bipolar patient and also some patient with schizophrenia. This GSK3 beta is a kinase that involved to the inflammatory response, and it also play very important roles in the like a phosphorylation, that post-translation modification of the SARS-CoV-2 proteins. Another mechanism is we call the CL-SY. One example is this BDNF. So BDNF is a growth factors that play very important roles in the neurotransmissions. And also it could target with many other proteins involved with this we call the mTOR signaling. And it shows that SARS-CoV-2 could enhance this mTOR activities. Because of this, then this virus factor could implicate in the mental disorders. The AC2 we talk about again that is in related to some anxiety or depressions, and also CMPR SS2 that is the host proteins that associate with some disease, depressions, and also cytokines that may associate with schizophrenia. So because in my research we always focus on these ones, we found the coronavirus could dysregulate the expression of these ones. This one could be impacted by the coronavirus infection, because it showed that this one they could reduce in the expression could reduce in the coronavirus infection, and it could associate with some immune dysfunction to the mental disorders. We not just show that this one, they are huge pathways. Right now, there are more than like 250 genes. One of the very important target is the NRX1. Like in the GWAS study, we show this target genes. So this NRX1 it could involve to the synapse formations, and also some expression could be changed in the bipolar or schizophrenia patient. So we also do some research for the NRX1. So here we actually research NRX1 AlphaFold models. AlphaFold is some machine learning method that we try to predict all possible protein structures in human and other organisms. Actually, we compare this model to some experiment structures, and it showed these two structures are very similar. Okay, RMSD only like 1.6. And also then we apply the computational saturation mutagenesis to look at all the key residues that may affect the protein stabilities. Then we found it work in the both computation model and also in the human experiment structures. So in here we try to use different bioinformatics to look at the pattern play very important roles in both COVID and also the mental disorders. Okay, so here is my conclusion. So we have three parts. The first part is we try to talk about how we sequence the different pathway genes, and we find the mutation in the NRX1, and also we find some signals in the different regulums. Then the second part, we use different bioinformatics to look at the coronavirus spike proteins. We found some key residues are important for the protein stability or binding affinity. And we also found some very interesting, like D684G, they could stabilize the spike protein, and also the L452R that could enhance binding affinity. That make us think about we could make some prediction method to predict some dominant strengths. Then the third part, I talk about the relationship between these two diseases. I think because we think that our bioinformatics is very useful, actually this is our future directions. So we try to make this viral mutation analysis platform. Okay, that is this platform actually we combine the we have the collaboration for them from the different part of our university. We work with Dr. Johnson, that epidemiologist, and also Dr. Teng, biologist. So we try to combine these different studies from the bioinformatics from biology, and we try to develop this kind of platforms. This platform could help us for the community monitor and also like give some COVID-19 alert and also help us to prevent some severe COVID, especially in our minority populations that could help us to reduce the health disparities. So this also need a community engagement center to do that from like Dr. Williams' support. So then I want to thank all my lab members, especially I think the two recently graduated Dr. Rhoda Zarina. So actually she is the first author of this paper at work on NIX1, at B work on SARS-1, and Vic work on the ACE2. And I think I collaborate with Dr. Teng for many projects for this coronavirus, and we are continuing to work on that. So as I showed in my lab, we use these 3 different mass next-generation sequencing machine learning structure modeling to research all this kind of disease, COVID-19, psychiatric disorder, and also other mental disorders, and also even the cancers. So any questions?
Can I ask a question, please?
Yes.
This is really very nice talk, very comprehensive, very illuminating talk. And I'm really surprised that, sorry, my video doesn't work, so I couldn't show my face. Do you hear my voice?
Yes.
It's really incredible that mental disorders such as schizophrenia are really more heritable than breast cancer, as you alluded into.
Yeah.
And it's also very surprising that despite being less heritable, breast cancer can easily be diagnosed at molecular level. I'm just wondering if there is any molecular approach looking at key genes, detection of key genes, and ultimately then to really diagnose based on that approach. Is there anything in that regard?
The key gene for the psychiatric disorder, right? You mean? Or cancer? Yeah. Yeah, Dr. Badada, so.
Yeah, and so you also alluded into your structural modeling, the structural modeling using the structural modeling and bioinformatics to really predict the effect of mutations. So if mutation happen to be stable, then it's going to favor the virus, to make the virus fit. If it is destabilizing one, then it's of course leads to the virus works against the virus, right?
Yes. Yeah. To answer this question, so I think during our research there are maybe 2 favorable things. First is if that mutation happened like this 4G that makes the spike protein more stable, that is favorable for the virus. Then the second is, of course, if some mutation could enhance its binding affinities, that could favorable for this virus host interaction. That also the very favorable for the virus. Then for the first question, I don't know because using next generation sequencing, we can sequence all this kind of the genetic variant, not just the schizophrenia, but like some breast, like you said, breast cancer, they may work like sequence the somatic mutations that could also help to identify some target associated with certain cancers.
Yeah, this is really very, very exciting. I have one more question. So as you know, currently BA5 is the dominant subvariant of Omicron, correct?
Yeah.
And is there anything known, at least from structural modeling, such as the one you use, that really implicates this virus to being why it's dominant? Is there anything known?
Yeah, actually we try to work on that because Omicron is kind of very different with the other like a Delta variant, so it accumulate many kind of this mutation. We don't know why this happened. As then we try to not just look at some single mutation, we try to combine the different mutations. And we find some combinations, this could enhance this protein stability or maybe enhance this binding affinities. So that is, I think we work with Dr. Teng and also Dr. Johnson to see, okay, if this kind of we can when we have the bioinformatic result, maybe we also think about some virology experiment to validate that. So that is why we try to develop this kind of the platforms. So hopefully in the future we can use this platform to predict this kind of the dominant virus strand.
Yeah. Okay. Thank you very much. Just before I let you go, one final question. We have a paper review, submitted, and so the reviewers, asked us to include some structural modeling regarding the different variants. And I didn't know that, there is, an expert, in the structural modeling in that regard. And I'm just wondering, I think the world is too small, this, seminar happened to really introduce me to you, where, we are really looking for people who could help us in structural modeling.
Yeah, of course.
And so I would really like to contact you and discuss with you. So this is, the review is really favored. It's just that, they want some structural modeling included. And I will contact you, and if we, working together in this, I will also include you in this review. I think if we, I get your email, maybe I can get it through Elisha, since I have her email, or I think this is I will use this opportunity in this regard. I mean, that's in the end, the purpose of all this meeting, to collaborate, to facilitate, exchange. And I'm really glad I happened to attend this meeting. It's very, very exciting, very illuminating talk. Thank you very much.
Yeah, thank you. Yeah, I think that is the purpose of the talk, so we can find many collaborations. Yeah, and just put my email address in the chat box. You can definitely.
Yes, I see. Yeah. Okay, thank you so much.
Yeah, I will also make sure that you will. I can do an email introduction with the both of you. I have Mr. Badada's information as well. Thank you so much. Sure. And I believe Dr. Teng has a few questions. He has unmuted himself. Hi, Dr. Teng.
Yeah, I can ask myself. Yeah.
Great.
Excellent presentation, Dr. Teng. I was a little bit late for the first part of your talk about the schizophrenia, the protein DISC1. Since I also have collaboration with Dr. Sava in Johns Hopkins, and he's working on the schizophrenia.
Oh yeah, I know.
Yeah, for a long time.
Yeah.
And one virus was involved in this DISC1 is our cytomegalovirus. And since DISC1 complex with PML, and cytomegalovirus can disperse the PML. So that's how the cytomegalovirus infection could be related to the schizophrenia schizophrenia. So I wonder, for the SARS-2 protein, for the SARS-2, which protein is related to the DISC1?
And that is, I think I need to do some search.
Okay.
So like that is one way. I think the one way related to these two diseases, we most focus on these DISC1 pathways. Like for example, like some that is, I think I'm working on that for some pathway analysis, how to build the, for example, this one, the AC2, the NRX1, how they can build together. And also that potential, I think maybe when we get the if our project are funded, then we can get the sequence from the COVID patient, then we can specifically look at these AC2 mutations, these one mutations, to think if they have some kind of relations. So this is the quick question we should answer in the future.
Okay. My second question is related to the SARS-2 evolution. So we say that SARS-2 is experienced very fast mutations from the original one to Delta, Beta, Alpha. Now is Omicron. Omicron is also changing.
Yeah.
Before that, we have very high load. The reason why for the virus to have so fast transmission is the virus can produce the high viral load, so it can one patient can spread a lot of virus in the air, then it will be transmitted to many people. So that we make a high R0, and so basically the spike protein is very important, especially the the original one to the Delta is D614G mutation. And then now for the Omicron, that's more than 30 amino acid changing. So that's why there's some group to try to make the Omicron a different virus, say SARS-3.
Yeah.
But, I wonder, you checked the affinity and the stability of the spike protein based on single amino acid mutations. I wonder if you have done compare the single mutation with the multiple or double or triple or multiple protein amino acid mutations. For example, the D614G mutation must increase already the spike protein stability or affinity. How about based on the D416G and then plus other mutations, then you compare to the D614G alone to say if there is any even higher and higher the affinity or the stability. That will be also important to explain why the one virus, once they are circulating, then they become dominant.
Yeah, actually, this is a great question. Actually, that is the Omicron present first time we will see this crazy accumulation from the, like 30 mutations. That I think I tried the different combinations of these multiple mutations, and we found that in certain combination, actually, it could provide more stabilities and also like more binding affinities. I think probably in these 30 mutations, some mutation maybe not so important. It's just randomly maybe, but in several key mutations, when they combine together, they may enhance this kind of the fitness or transmission rate. So
yeah,
this is this is why interesting. We want to see what kind of the mechanism behind these kind of combinations.
Yeah, my last question, maybe not a question, maybe a comment, that is your post-translational modification program. that will be also important because the S protein, spike protein, usually help the virus infection by fuse the infected cells with the uninfected cells. Then this virus transmit from infected cell to the uninfected cells, then make more and more virus. And that is really the case in the lung tissue.
Okay.
Once the virus, even with a very low load in the lung cells, then if the virus want to make more, then they have to fuse the two different cells and make the higher viral load. So that would be also important. Then maybe we should check the mutation of the amino acid could help the spike protein in the change their modification, post-translational modification. That could be make more sense in the pathogenicity.
Yeah. I think that's a great suggestion. So, basically, in our lab, we had two directions for the post-translational modification. The first is we try to develop some machine learning tools for that. Because before that, only like for general protein, but how about the virus protein? That could be very interesting. Then the second, like Dr. Teng suggested, so if we apply some tools to predict this mutation may be involved to the post-translation modifications, then we definitely should do some experiment. And this could, hopefully, we can find some reasons so why this mutation are very important to the virus functions.
Yeah. Okay, thanks.
Are there any other questions?
Can I ask one question? Just using this structural modeling, is it possible to really predict before any mutation occurs in the future? Okay, before any mutation, before a new strain strikes, is it possible to really predict? This is the likely mutations that's going to cause pandemic. That's going to really be the dominant strain. Can we really say this in advance so that we can take some preparatory actions based on that information? Is it possible as based on current knowledge?
Yeah, I think that it's possible, and also we try to develop this kind of tools because use the computational saturation mutagenicity, we can look at all effect for all possible mutations for their stability and the binding affinities. Okay, so we want to know, okay, if some mutation do have these functions, is that very important for the future dominant strands? So this we need to know, if, for example, maybe by some combination of these mutations, or maybe this mutation could involve to any other functions. If involve that, then they may make very important. So yeah, I think that is basically the possible. And also I know some right now some group use deep learning, like one of the machine learning methods, try to do this kind of the work. So that is also our work, how we could involve this machine learning, combine the machine learning and also structure modeling some features to make this kind of the predictions.
Very exciting.
Yes, I actually wanted to add to that question. I had the same question, and I was also wondering if I'm not as familiar with the research, but would it be possible to use the mutations that you're talking about, the ones that destabilize the spike protein? Would it be possible to use that as targeted therapy for either vaccinations or future medications?
Yes, that's great to talk. Actually, we and Dr. Teng, actually we try to develop some therapeutic peptides that not the spike, but we focus on use the ACE2, that is a receptor of ACE2. at right now and dot time, we try to look at the activity of all these pipette. Hopefully, we can develop some antiviral for this.
Yeah,
facility. Yeah,
that's exciting. I can't wait to hear what you all find. Well, if, are there any other questions from our audience? Okay, well, thank you so very much, Dr. Teng, for taking the time to tell us about your research and answer questions. Thank you to everyone for joining us and participating in the discussion. I would like to invite you to our next journal club. Let me share my screen. To our next journal club. The bad of smoking, but the potential good of nicotine in COVID-19, featuring Dr. Yusuf Tizabi of Howard University College of Medicine, Department of Pharmacology. This will be on Monday, August 29th at 12:30 p.m. You can register by clicking the link that I put in the chat, and you can also email us at rcmi-cec@howard.edu with any questions about today's presentation or to learn more about how we can support your research projects. We will see you on the next journal club. Take care. Thank you, everyone.
Thank you very much.
Thank you.